Thank you very much, Mr. Chairman. It's really a pleasure to be back. I was here last year, and as you say, we met in Leiden, and I now have the feeling that we are in a group of friends, and that's really very much appreciated. It's a fantastic meeting. It's really my first typhoon in a Tanabata festival. It's great. I would like to switch a bit the topic to immunology and chronic inflammatory diseases. They have been mentioned a number of times. It's a problem when the immune system attacks our own body. Inflammation is something which needs to be under strict control. And the last discussant brought already up the idea of Tregs. That's a sort of new invention by immunologists. There's a cell that specializes in controlling inflammation. And my talk is really going through the things with Tregs. So the main question in immunology, how is this system organized? How these Tregs know that they have to control inflammation? And there the heat shock proteins come up. So the first thing that brought me into the field of heat shock proteins was this paper when we were working together with our friends in the Weizmann Institute in Israel, and we cloned an antigen from mycobacteria that we could use to manipulate arthritis, an experimental model of arthritis, and we could, as it were, we could vaccinate against arthritis. The funny thing of this paper is that the word heat shock protein was not mentioned. But when we found out later that it was in fact a heat shock protein that we had cloned, HSP60, it was referenced a lot, and that's why I bring this up. But in the bottom you see that we are manipulating a model of arthritis with just a single epitope, with a peptide derived from a heat shock protein. Well, just to give you an idea of heat shock proteins, they are families of molecules which are essential in life. They are mainly their main function is protein folding. So you see a number of these families. For me, HSP60 and HSP70 are the most interesting heat shock proteins. I think they are the most relevant also for the maintenance of tolerance in the immune system. You see here that they are active as folding catalysts in many cases, so they are also called chaperones. They interact with other proteins. And just to give you an example, for HSP60, for instance, HSP60 is a chaperone, and it is one of these building blocks. There are 7 of these HSP60 molecules in a ring structure. So it's a 7-mer, and there are 2 of these 7-mers stacked upon each other, and there is another cap on it, HSP10, and it closes. It's sort of organelle in the cell. So bacteria have this. This is GROEL, that's in E. coli, the structure that does the protein folding. But also in mammalian cells, we see the same thing happening. So here is an unfolded or badly folded protein. It comes into this structure. It makes transient interactions with the inside of the structure. And when it's properly folded, it gets out again. And this is all it does. So it's a tricky business for these proteins, and that's probably the reason that they are so evolutionary conserved. So if you go to bacteria, or you go to plants, or you go to humans, you will find the same proteins, same structures. And the main question now is, what do they do to these famous regulatory T cells? So these cells have their profession is to control tolerance, to control inflammation. These are the housekeepers of inflammation. So the question is, if these proteins are being targeted by these proteins, how does this work? Well, first of all, they are evolutionary conserved. So if you go to bacteria, we are repeatedly interacting with bacteria, for instance in the gut, in the microbiota, we see these proteins constantly, and we try to react against the conserved areas of these proteins. Then the second thing is that they are the most frequent cytosolic nuclear MHC class II natural ligands. So if you go into the immune system, T cells recognize antigens in the context of so-called MHC, major histocompatibility complex molecules. And Tregs recognize antigens in the context of MHC class II molecules. Well, if you dig into these grooves of these molecules, you will find very frequently parts of heat shock proteins located there. So the immune system constantly sees these proteins. And if there is inflammation, there is upregulation of these proteins. That's why we call them stress proteins. When there is stress in the tissue, you get upregulation of these proteins. Well, when we had found the activity of these molecules in adjuvant arthritis, many people used these proteins in arthritis models, but also in other models like diabetes, atherosclerosis, EAE, which is a model for MS and allergic conditions like asthma. And there are various of these molecules used, mostly HSP60 because that was the first molecule that we cloned, but later on many other of these family members of the HSP families were used. And what they had in common is that they protected against these experimentally induced models. And then Steve Anderton, he is an immunologist now in the UK. He came to us on a fellowship for a couple of years, and he did a marvelous job. He cloned the T cells in our arthritis model, and he showed that these T cells were responding against 8 different epitopes in the HSP60 molecule. And there was one epitope that stood out because that was a very conserved epitope, and the T cells were cross-responding between the mycobacterial sequence and the mammalian sequence. So the cross-reactive T cells were there. They could not distinguish between bacteria and human heat shock proteins. And he showed that this cell could protect against arthritis, and that also this epitope in the form of a synthetic peptide could protect against arthritis. So this brought to us the idea, yes, it's the cross-reactivity between microbes and the self that contributes to the maintenance of tolerance. We can inhibit arthritis with these T cells and also with these epitopes. All the others did not. There were some epitopes even making things worse. So this is severity of arthritis in these curves. So there we had the idea, and that's here. So microbial heat shock proteins are very immunogenic proteins. If you think of HSP60 as the major antigen, if you take coli bacteria and you immunize a rabbit, the first thing the rabbit does is make antibodies against HSP60. So the immune system likes to work with these proteins. So that's probably for the purpose of pathogen elimination. So the immune system reacts against the non-conserved epitopes in these molecules. But when it responds to the conserved epitopes, it drives a regulatory T cell, which is cross-reactive with the self, and that is contributing to tolerance against inflammation. In another way, you can say there are in the immune system, there are cells that see these proteins. When there is inflammation, in this case in the joint, these cells go there, they see the cross-reactive target epitope in the inflamed tissues. It's stress, it's inflammation, there is a lot of stress in these cells, they produce these proteins. So these cells recognize this, and this is how they can contribute to the control of this inflammatory process. And so the system can be manipulated in various ways, so you can inject these heat shock proteins or their peptides in order to drive the system and to vaccinate against this problem. Or you can think nature does it by infection. So if we experience infections, these bacteria go in. It's similar to what happens in the mucosa, in the gut. We are ingesting, the dendritic cells are taking in microbes from the microbiota, and they show it in the mesenteric lymph nodes to the T cells. And so this happens continuously, but it is for these bacteria stressful to become here inserted in these cells. It's stressful for bacteria, so they produce their stress proteins, they produce their heat shock proteins, and so for the immune system it's easy to see these proteins in these situations. Here you may think of, for instance, the hygiene hypothesis. If we need infection, we need contact with microbes in order to organize our immune system. This may be an example of the molecular basis of this hygiene hypothesis. So it's a fundamental process. It's not only arthritis. So people have been wondering about atherosclerosis. It's very interesting that if you look for the T cells in atherosclerosis, you find many T cells responding to HSP60. This is just an editorial in one of the top journals on atherosclerosis, where Göran Hansen is just thinking about making a vaccine against atherosclerosis using these heat shock proteins. Some people have made their business out of it for a lifetime. For instance, George Wieck in Innsbruck in Austria. He worked for many years on atherosclerosis and HSP60, and now his daughter-in-law here, Cecilia Wieck, she is also continuing with this activity. Just to give you another idea of what is possible with these proteins, this is a group in Brazil. We are collaborating with them, and they used a peptide, B29, which is an HSP70 derived peptide. It's also a conserved peptide. It's from the mycobacterial HSP70. And when they administer this to mice, they can do transplants. Here you see Balbc mice, the white fur of a Balbc mouse, but they transplant here a C57 black skin. And that's a huge difference genetically. So mostly these skins are rejected very early. You see this happening here in the controls. But if you pre-treat these animals with HSP70 and this particular epitope, you will see that the skin stays. So this is for a longer period of time. You see here the black skin on the white fur of the Balbc mouse. So you can manipulate tolerance not only in inflammatory diseases but also in transplant, and that's what they are aiming for. We have been looking into patients with autoimmune conditions a lot in Utrecht, for instance, in children with juvenile chronic arthritis. And that's an interesting disease because a subgroup of these patients remits the disease spontaneously. It's still an aggressive disease, but they remit. And so we are interested in knowing how the immune system is going to remit, what's going on there. Well, if you then monitor the T cells, you will see that during remissions, and especially in the patients that do remit, you see these responses against HSP60, and a number of people have made their PhD thesis out of that. A similar thing may be ongoing in MS. This is in the relapsing remitting MS. There you see antibodies against HSP70, which is not present in the patients with progressive MS. This is from the Weiner group at Harvard University in Boston. Now, these stress proteins are for some people, they are typically the molecules that induce danger to the immune system. They are sort of alarm signals. It makes sense. There is stress, so you need to alarm the immune system that there is stress. And Polly Matzinger, who is here on the photograph, she is the proponent of this idea of danger to be very important for the starting of the immune response. This is Jeff Bluestone. He was the director of the American ITN, which is the Immune Tolerance Network. So they are spending money in organizing clinical trials on inducing tolerance. And this is our PhD student, Martijn van Herwijnen, discussing his poster in a meeting. But these people, so he is the man of tolerance and regulation, and she is the one for inducing danger. So these are totally different views on the problem of heat shock proteins. And so the field has been actively working on this, and there is some contamination in the field because of the fact that these recombinant proteins were contaminated with endotoxins like LPS. And that, of course, activates antigen-presenting cells in the immune system. So if you take away these contaminants, you don't find this. And we have been trying to summarize what's there in the field. So we think they are not too much of, they are not damps, they are just dampers. They damper the immune system, they help the immune system to control and to be tolerant. Since they are the damps, the true damps are intracellular hidden proteins, HSPs are sometimes extracellular, so I don't think that they can be real damps. They protect in these experimental models of autoimmunity, and dendritic cells become tolerogenic when they are exposed to these proteins. And that has been noted by many people. This is a publication from the Bonorino group in Brazil. They see that Mycobacterium tuberculosis HSP70 impairs the activity of dendritic cells. They are going to produce interleukin 10, which is a cytokine that is actually anti-inflammatory. We ourselves have seen similar things. So we have incubated dendritic cells in the mouse system with mycobacterial and mouse HSP60. You see that they are then becoming modulatory. You can transfer these cells into the model and they suppress arthritis. So these things are much in line with the idea that stress proteins are in fact helping tolerance and not so much danger. Well, I mentioned the HSP70 epitope B29. This is a mycobacterial epitope, so it's microbial, it's derived from HSP70, and we work with this peptide in BALB/c mice. And in the BALB/c mouse, you have a very interesting mouse experimental arthritis model where we immunize with proteoglycans from human origin. We immunize twice, and we get a relapsing and remitting disease in these mice. If you treat them with such a peptide intranasally, so just it's inhaled by the mouse, part of it will probably end up in the lungs. But then, so you approach the mucosal immune system, and what happens is that you reduce arthritis. This is the bacterial peptide. It reduces the severity of arthritis. If you take the cross-reactive epitope, so if you take the murine or mammalian homologue, you can see similar things. So this is in line with what Steve Anderton saw with HSP60 in the earlier studies. Well, Martijn van Herwijnen, that's the PhD student that was on the slide, he immunized mice with this single peptide, and he took the regulatory T cells. They are in the CD25 positive T cell population. They are our regulators in the T cell system. And so he transfers the cells from these immunized recipients into the mouse model, and then you see that you can prevent arthritis. In this case, he used 1 million of these cells. That's not much if you compare to what other people do with other systems. And you can also drive therapy. You can also induce therapy. So you wait until the animals develop disease, and then still you suppress disease with these cells. So these are the cells that do the job. These are the anti-inflammatory T cells, and that they are really actively reducing disease is visible in this slide because we have here used a monoclonal antibody against these cells. There's a genetic marker on these cells that were transferred, and this monoclonal antibody depletes these cells in vivo. So here the cells are suppressing disease. We give this monoclonal antibody and we see the disease comes back. So the cells are actively controlling disease. Okay, as I mentioned, there are a few questions in the field. So how can these cells be so dominantly active in inducing tolerance? There are a few arguments to make here. The first one was that they are evolutionarily conserved. You see this here. These are HSP70 sequences of mammalians. This is E. coli and this is Mycobacteria. And you see here, for instance, this is the B29 epitope, the huge level of conservation. All the amino acids in black here show that they are identical. So it doesn't matter whether you go to a mammalian HSP70 molecule or you go to a bacterial HSP70 molecule, you will find stretches of amino acids which are completely identical between these molecules. And for the immune system, it's no problem to respond to them. So the T cells respond to an epitope like this one, B29. So then, what does that mean if they are so conserved in bacterial species? So as I mentioned already, we are in contact with the microbiota. The microbiota is taken in. And so in the field of immunology, there is the question, how do the regulatory T cells here perceive the presence of these proteins in these bugs? Are these T cells coming from the thymus? The T cell is named a T cell because of the fact that it is educated in the thymus. Is it coming from the thymus? And then does it have a relatively unique nature when it is here in the colon? That's one view. Another view is that the T cell receptors are shared between the thymic and the colon T cell receptors. Well, this is in discussion, but in both cases, it must be so that there is contact with these bacterial or food antigens in this area where it's the main place for T cells to reside. So here, when they are shared between thymic and colonic, so the T cell receptors are shared between these populations, then you can take it for granted that in the periphery, they recognize cross-reactive microbial antigens in the intestine. And so by the repeated contact with these microbial antigens, we set a focus on the conserved parts of these molecules. Then their frequent presence in the MHC class II molecules. This is based on many observations. This, for instance, is a key paper for us. It was a science paper in 2005. You have to go to the supplementary data, and there you will find this table. And the table tells you that HSP, so this is the heat shock cognate protein, that's an heat shock protein 70 family member, HSP17 itself. They are the most frequent cytosolic nuclear MHC class II natural ligand sources in combination with another one. They are relatively long-lived proteins. They are loaded in the cells and there must be a sequence in the MHC molecule because otherwise the MHC doesn't reach the surface of the cell. Well, still intriguing, but we don't understand it completely, is that the HSP70 molecules are being coded within the MHC. So it seems like evolution takes care of the fact that these are evolving together, the MHC and the HSP70 molecules. Maybe because they both interact with other proteins. They are both sort of chaperones. Now let's go to the thymus. So in the thymus, the T cells are being educated. Which antigens do they see in the thymus? They see self-antigens. We educate all our T cells on the basis of recognition of self. That's one of the things that we know for sure. So then how about these stress proteins? Well, if you go to this paper, you will see that the stress proteins are here in green, and they are preferentially found in the thymus populations that are expressing HLA class II molecules, MHC class II molecules. Here they are, on the dendritic cells and also on the cells that are depleted for the dendritic cells. There are even more. So there is plenty of opportunity in the thymus to present these stress-related proteins to the T cell system in order to make sure that there are these cells around. And if you go to the table in the same paper, you will find here that our peptide here, the B29, is also present, being found in the dendritic cell populations and also in the depleted populations for the dendritic cells. So also in the positively selecting populations, you'll find a peptide like the B29 epitope. And other people have made this sort of very nice pictures of the histology of the thymus, and they see also HSP70 as an example molecule in the thymic epithelial cells. So there's a complete concordance between the presence in the thymus between these molecules, HSP70, and the cells that are taking care of the positive selection of the repertoire. If you go to the dendritic cells, you will find that there is almost no HSP70 there. It's maybe difficult to see. You have to go to the paper really to see it. Then the upregulation when there is inflammation. So we like to go to arthritis in these cases. So this is the example of rheumatoid arthritis. So this is the synovium, the inflamed synovium of rheumatoid arthritis. You see HSP70 there present in these inflamed joints. If you go to oligoarthritis, if you go to you know, the chronic osteoarthritis, then you don't find much HSP70. So this is a less inflammatory disease. It has an inflammatory component, but it's not like rheumatoid arthritis. And if you take the cells from the synovium, you expose them to HSP70, you see, or you expose them to raised temperature or to TNF-alpha, you will see the upregulated presence of HSP70 and also of the heat shock factor. It's also the transcription factor for these molecules. It's being upregulated when there is stress. So the stress can be heat or can be inflammation. Both drive this system. Then this paper, this is from Hans-Georg Rammensee. He is one of the pioneers already for a long time in showing us what is in the grooves of MHC molecules. And this is a very nice paper because he is here putting stress on the system that leads to autophagy. So when cells are under stress, they are going into a process that the cell biologists call autophagy. So the cells internalize the internal proteins and use these proteins for making up their own novel proteins. When he's searching in this system in human MHC class II molecules for the presence of peptides, he finds again, no surprise, HSP70. A lot of HSP70 molecules are there. And we were quite excited to see our B29 epitope also showing up here. So this is a human HLA-DR4 molecule on B cells. HLA-DR4 we know is associated with rheumatoid arthritis in humans. We see our peptide there in the groove of these individuals with HLA-DR4, and it's exactly this mammalian homologue of this mycobacterial epitope. So it's there already in the MHC class II molecule. Well, lastly, to give you an example of how these stress proteins can inhibit inflammation, I would like to show this paper. HSP70, you probably know these people, they are from Japan. HSP70 mediates also the degradation of this inflammatory subunit, NF-κB. It was mentioned already by Nancy this morning. NF-κB is the major inflammatory mediator, and these proteins interact with the production of NF-κB at various levels. There are many papers out in the literature. So to summarize this, they are evolutionarily conserved. It's probably the major thing in the fact that these proteins are so involved in tolerance. They push regulatory T cells at the level of the mucosa. We end up with mucosal tolerance. It may have a bearing on the hygiene hypothesis, as I mentioned already. They are the most frequent cytosolic nuclear MHC class II natural ligands. So the immune system sees that they are there. It's a sort of default recognized system. They are the default antigens for regulatory T cells, you can say. They are upregulated during inflammation, so it targets these cells to the site of inflammation when it is necessary. And so it seems like that cell stress is a physiological trigger for this sort of regulation. Also the inhibition of NF-κB plays a role there. And also IL-10 is known to be a stress-associated cytokine already for many years. So this is one of the papers proposing that cells under stress do produce this anti-inflammatory cytokine IL-10. So stress proteins and tolerance and the anti-inflammatory effects have something together to say to each other. So now I come for a brief series of slides on the food, the side of the food. So we have seen inflammatory stress upregulates this protein that can drive regulatory T cells. I've showed you that in the model of arthritis. There's one funny thing. If we age, if we go to the elderly individuals, we will find that this system is not more so responsive. That's maybe one of the factors leading to the fact that in elderly individuals we see more inflammation. So we are looking for compounds to improve this system. And they are known to be co-inducers, so they co-induce the expression of these proteins when there is stress. We are not looking for toxic elements that are stressful to cells. No, we want to have proteins or elements or compounds that drive the presence of these proteins when there is stress. So we are looking for co-inducers in order to help in this situation when the system is not responsive, that responsive, to improve this system. So this is the inflammation. We know there's low-grade inflammation under aging. We develop, that's one of the problems of aging, we have a sort of low-grade inflammation that we try to control, and in some cases we are not capable of doing that. So to bring this in a sort of hypothetical figure, so the regulatory T cells see these proteins in the context of the MHC class II molecules on the antigen presenting cells, and you can manipulate the system with these mycobacterial peptides like B29. You can do the same with the self-peptide. I showed you the mouse model where we gave the peptide intranasally. But you can also think of upregulating the heat shock proteins in the cells by, for instance, using these co-stimulatory, co-inducing compounds. And that is what I would like to show you that may work. We have here one example. This is oregano. This is a PhD thesis of a student that's already for some time ago. She discovered the antibacterial activity of essential oils of oregano. And essential oils of oregano consist of carvacrol. And we were wondering whether carvacrol was also doing something on mammalian cells. And we're very lucky in finding that if you incubate cells for a couple of hours with this essential oil of oregano, you will see that nothing happens at a normal situation of 37 centigrades Celsius. But if you increase temperature, then in the presence of carvacrol, you will see an upregulated presence of HSP70. This is typically the effect of a co-inducer. So you need stress, in this case heat, in order to upregulate these proteins, and then the carvacrol helps the cell to produce this protein. That's co-induction. And that works in mice, and it works in humans. And if you give this carvacrol orally by intragastric gavage in mice, you will see in the Peyer's patches, so in the lymphoid organs of the immune system, you will see the upregulation of HSP70. The message for HSP70 goes up. And also T-cell responses can be detected in these mice. But the main thing I would like to show you here is that it suppresses arthritis. So this is again this proteoglycan-induced arthritis model. Proteoglycan arthritis in Belpsy mice. They have received this carvacrol just from oregano orally, and it's almost impossible to induce arthritis. It takes time, and then they develop some arthritis. And you can transfer this effect with T cells. So this is a T cell mediated anti-inflammatory effect. If you take the T cells from these mice, you transfer them in other recipients, healthy recipients, you induce arthritis, you will see it becomes more difficult to induce arthritis. So it's a T cell mediated phenomenon, and it's just resulting from the eating of this essential oil. So food components can boost the protective cellular response to these proteins. The immune system transduces its information into regulation and suppression of inflammation. What we eat can upregulate immune regulatory T cells and downregulate disease. And this is again a nice example. So this is carvacrol. It's a very simple molecule, but the mouse doesn't like it, and humans don't like it. It smells. It's not. You need quite a lot to. You want to cure your own arthritis. I will not do it with this carvacrol, but it works. It's very impressive. So these proteins, they control inflammation, but also they are involved in the reorganization of the epithelial barrier. And that's another thing, I have no time to discuss it. But the They assist, for instance, the cytoskeleton, the F-actins that are important for the intestinal epithelium integrity, but also the tight junctions between the cells are being improved in their barrier function by these proteins. They are chaperones. Other proteins, in order to optimally function, they are helped, they are assisted by these stress proteins. So if we co-induce these proteins, it's good for our barrier function in the gut. And this is just an example of a paper that is addressing a very nice finding, and that is that this is in a rat system, that mother's milk in these animals increases the presence of these proteins here in brown in the gut, and it also improves the barrier function. So you can show barrier function with labeled dextrans, and they go in the controls. They go well through the barriers, but if you have fed these mice with mother's milk, it doesn't work. So there is something that is present in mother's milk, driving the presence of these proteins also in the gut of the just born animals. I'm going to conclude with a few remarks. So we are searching for these co-inducers in food. We find them by taking cells and looking for the intracellular presence of HSP70 in the cells. We have developed a system of reporter cell lines for semi-high throughput screens for these molecules. And we collaborate, of course, with industry, both for animal nutrition, but also for human nutritional purposes. This is just an example. This is, for instance, black tea. I think it was already on the slide of one of the previous speakers this morning. Black tea is one of the materials that has the capacity to co-induce the presence of stress proteins. We see it here, black tea. We give these extracts and we see here HSP70 going up. The same is true for saffron, for instance. And here is again carvacrol. So these molecules do this, they upregulate. We were collaborating in that case with an Italian group, and they organized a few years ago a very nice meeting, Eating for Preventing Inflammatory Diseases. So you can, I think there is a booklet with proceedings. You can find all that was presented in that very nice meeting. So this is our high throughput screening system. It's a luciferase system, so it's a standard system, but we can now discriminate between various promoters that are being induced for the HSP system. And this is again carvacrol. You see that in the presence of arsenite, in this case arsenite as our stressor, if there is low degrees of arsenite, we don't see anything of these co-inducers, but if we have higher arsenite as a stressor, we see that with the presence of carvacrol, HSP70 goes up. And so for Utrecht in our university, we have a focus now on food. So we optimize these systems to find these HSP co-inducers. We determine the HSP co-induction capacity of, for instance, vegetable varieties. We are very interested in tomato at this moment. We will develop a program on this. And we assess the differential accumulating metabolites in these fruits. So this is a collection of people that did the work. Thank you very much for your attention. And I'm very happy to answer questions.